Good evening, everyone. Thank you all for choosing NIPOMAN, NGS, and RNA-seq internship program. I'm sure for few it will be a repeating session, but many have joined us for the first time. So I kindly request everyone to pay the close attention throughout this program. If you have any questions or any doubts, please feel free to unmute and get clarified or post your queries in the chat box. To support your learning, we will be providing you with the session recordings. Recording will be uploaded in the Google Classroom. I will be sharing you the link shortly. And also study materials will be shared through the Google Drive. I think this is clear. Without further delay, I would like to introduce Ms. Anshu from IBAB and also a distinguished NGS expert at NIBOMAN. She will be guiding and mentoring you throughout the internship program, helping you to build both theoretical understanding and practical skills in NGS and RNA-seq analysis. Thank you all, and I wish you have a productive and enriching learning experience. Over to you, Anshu. Please take over.
Thank you, Pavani. So again, a very good evening to everyone. So I'm Anshu, and I'll be taking the NGS and the RNA time period. So without any delay, I would like to start with today's session. I'll just present my screen. Is my screen visible?
Yes, it's visible.
Okay. So today I will be discussing majorly about RNA-seq analysis. And RNA-seq, like I've written as RNA-seq, it's basically RNA sequencing. So just we'll be talking about that. So RNA sequencing is one of the most important, and you can say one of the most used application of NGS. So NGS here stands for the next generation sequencing. Okay. So what basically RNA-seq helps us to do? So RNA-seq allows us to study if there is any gene expression that is happening at a genome-wide scale or in an individual, or if we have to compare it between individuals. So it basically helps us to understand that how active are these genes and how strongly are they expressed in a particular individual. So this difference in the expression between the individuals can also be recognized and understood by this RNA-seq analysis. So in this particular lecture, as this is the first lecture, so it is going to be a little basic. You guys might already know about it. It might be a repetition, but as it's the first lecture, so we'll start from the entire basics and we'll move further step by step. So in this lecture, we'll be basically covering the basic concepts of the transcriptomics, and we'll learn about the principles of RNA-seq and the workflow. So it will start with the DNA evolution. So basically, I'll be stating all the technologies that have been used from the past decade till now in the NGS technology and how these technologies have evolved with time. Because we all knew that an initial time point, getting a sequencing done, you all remember, right? The first genome project, that is the human genome project that happened in the first time. So it costed billions and millions and billions of dollars. So now, as the time is increasing, we can see that the sequencing is becoming more cheaper and cheaper because of the, I apologize, because of the increasing technologies and advancements. So we can, that is why we are, this pricing, the entire thing is also getting decreased because the people are more accessible to it and it can be done more easily. So apart from this, we'll be discussing about the NGS principles. So this NGS principle includes how this NGS sequencing is done and what are the steps that are followed, which will be followed by the transcriptomic concepts. That is about the RNA sequencing, the concept of transcriptomics. What is transcriptomics? What basically what is the factor that we consider transcriptomics? All of that. Then we'll move on to gene expression. So gene expression we'll understand how this RNA-seq data that we get from sequencing, how is this data used to measure a gene expression? After we understand that, then we'll have an analysis workflow. That is, I'll be summarizing, or maybe I'll be introducing the entire RNA-seq workflow from the starting step till the enrichment step. One by one, we'll go through all the steps and we'll understand that how these steps are working and how are they connected with each other. Apart from this, we'll be understanding about the major applications and then the advantages, disadvantages, and all of that. And in the end, I'll just mention about the project. So you guys know that after the training session where we'll be covering the entire RNA-seq workflow, then we'll have a small like a project, a mini project will be given. So what and all will be given in the project, what and all we are expecting from you, that and all also I'll be explaining a little about it today, and then later on whenever we move on to that part, I'll elaborate it more. So now we'll start with the basics. So why are we studying gene expression? The basic question when it comes to RNA-seq, right? So like, why are we studying gene expression? So every cell, like in an organism, basically all our cells in our body, they contain DNA. All this DNA perform function. Okay, so like there are certain cells that are expressed, or certain genes that are expressed in a larger amount, and otherwise other genes are expressed in a smaller amount. So to understand this level of expression, so which genes are expressed more or which genes are expressed in a lesser amount, we study gene expression. So for example, if we take that neurons conduct nerve impulse, and liver cells are basically involved in detoxifying chemicals. These are just basically two sets of genes that I'm just talking about. And there are certain other genes like muscles. They improve in, like, they're helping in movement. So the difference is not in the DNA sequence, because the DNA is going to be the same. The thing is at the expression level. At the expression level, the difference is happening. When we are getting converted into an mRNA and the proteins are produced, those set of things are different. That is what they're making them behave differently. So gene expression is basically, now like if we have to give a definition to it, so gene expression is the process by which the genetic information that is stored in the DNA is converted to a functional molecule like RNA and protein. And when we are measuring this gene, like when we are measuring this gene expression, that is when we are using this technology called as RNA-seq analysis. So when we measure and when we understand this gene expression, we can understand how the cells are functioning, and then how is the development happening of a particular disease, like are certain number of genes overexpressed or underexpressed, or what are the condition that is leading to this particular disease or this particular condition. Then how are these organisms responding to an environmental change? Because see, if a certain amount of protein, say like if I'm a cancer patient and another person is a healthy person, there are certain differences that might happen in the metabolism, because for my body, as I have a cancer, so that cells require metabolites as compared to the other. So there will be definitely a difference in the mRNA expression. Some genes that shouldn't be activated are activated. That is why the person has transferred to a stage of cancer, whereas those genes are like not active, they're inactive in a normal person. So because of this change in expression, that is when you can understand that there is some disease or there is a normal individual. So based on that, we generally compare about that as well. So if you have to take a real world example, as I already mentioned, that is a cancer cells. So cancer cells definitely express genes that are different, and based on this, they will have a different genetic markers. They will have certain markers that are expressed differently. So we can detect that. We can detect a specific gene. So say gene X, it's supposed to be like a normal expression gene. So, but when we are comparing with the cancer patient, that gene is activated, that is hyperactive, that is, it is expressing too much. That is why that particular person has been gone to a stage of cancer. Next, we can take in consideration as agriculture. So in agriculture, there are plant activated stress response genes that are activated during drought or during any calamity that can happen. So to protect that, there are certain stress response genes. Some genes get inactivated, because of which the plant is not able to survive. So all this comes in a connection. So there are certain genes that will be responsible for some stuff, and we need to keep sure that if they should be expressed in what they're required, the required amount. If it is more, upregulated; if it is less, downregulated. Both of them is going to cause some differences. Then next comes the medicine. So immune cells also alter gene expression during infection. Whenever we get an infection, there are certain proteins, right? Immune proteins that are produced more as compared to other. So that also correlates to the entire gene expression part. So basically, the main concept from here we should grasp is that the gene expression. What is gene expression? And how is this gene expression leading to a change in a human body or in an environment or to any subject which we are studying? So now, the foundation of molecular biology is based on the central dogma, that is from DNA to RNA to protein. So as you can see from DNA, it stores the genetic material, and then RNA, it acts as an intermediate molecule where it serves as a template for the conversion to protein. And then this RNA is converted to protein that basically tells what function is going to happen. So from a DNA, that is getting from DNA, which serves as the storage molecule containing instruction, and RNA is produced. And finally, a product, that is a protein, is produced that is responsible for all the cellular activities that is happening in our system. So since RNA molecules are only produced when the genes are active, we know that there are certain genes that produce, that get transcribed, and then only RNA is formed. So that genes are active, and that active genes produced RNA. Those RNAs are analyzed directly by checking how much protein they are producing, and that is how you can determine the activity of that particular gene. So this is why RNA-seq is important. So you have a gene, right? And now from this particular gene, an RNA is getting transcribed. Okay, so this was DNA, now we have an RNA. So now if, like that when you're replicating a DNA, maybe from one strand, one is produced. But if a gene is highly transcriptionally active, so if you have a DNA, it might have hundreds of copies of the same RNA because that gene is very highly active. Whereas, say, gene B, that is not that high active. So maybe that can have, say, 20 copies, but gene A is more active, so it has 100 copies. So you can understand by the amount of transcript that is getting produced that how highly active a particular gene is, and based on that you can get its sequence, and then you can analyze the data, and then you can understand. So at this current point we are at the pre-stage, that is we are trying to understand the genes that is present in the DNA, and then how they're transcribing it to like RNA, and then the sequencing steps. So first we understood what is this expression, what is this transcription, and why do we need all of that. Now we'll move on to the RNA. Now we'll move on to the part where we are also getting it sequenced. And today, for today's session, we'll only understand the entire workflow, and then in the coming sessions we'll be taking each and every step that I'll be discussing today individually, so that you can all perform alongside as well and understand why we are analyzing this and how we are doing the entire steps. So now the sequencing, as I
Anshu,
mentioned, yeah,
sorry to interrupt you. Yeah.
yes,
Before
yes,
proceeding, let's get a quick confirmation from our participants because they are new to bioinformatics. So
okay, okay.
I just want to know whether they understood the concept now.
Oh yeah, yeah. So are you guys
Yes,
able to catch up with
yeah,
me?
please go ahead. Can you please give thumbs up if you understand the concept now?
Actually, I won't be able to see thumbs up. If they can just unmute also, that will be fine because I'm at my presentation slide. So it will be easier for me
And
to
also, I
understand.
have a small request, Anshu. Can you please reduce your face because they are new to bioinformatics, right? Few terms might be
Okay, okay.
new to them. So I'm just
Yeah,
requesting you to slow
yes,
down
yes.
the face. Yeah.
Yeah, definitely.
Participants, can you all please give thumbs up if you understand till now? If not, if you want to repeat any concept also, please let us know.
And guys, if you have any doubt in between, if you think that I'm going very fast and you need any clarification, you can just unmute yourself, stop me and ask the question. I might repeat, like I will repeat as well. So
Sure.
that's not an issue. That's fine. Like,
Vafa
okay.
has raised a hand. Vafa, do
Yeah.
you have doubt, any doubt or it's by mistake?
Sorry, ma'am, it's by mistake.
Oh, it's okay.
Fine.
Okay. That's, yeah. So if you guys have any doubt, don't hesitate. Just unmute yourself and ask me to stop so that I can explain it back again. Because this is the basics. You need to understand the basics. Then only you'll be able to understand properly the further parts. And if you think I'm going fast, you can just tell that please go slow. And I'll try to go slow now. If you still think that the pace is a little faster, then you guys can still tell me. Okay?
Fine, Anshu, let's proceed.
Yeah. Okay. Okay, so I was here. So evolution of the sequencing technologies. So as I mentioned before, that that initially when the human genome project was carried out, okay, it was done using Sanger sequencing. Sanger sequencing is a very basic technique that was evolved in that time, and that has been like that was the origin of the sequencing thing. That is why we called Sanger sequencing as the first generation sequencing. So in Sanger sequencing, what Sanger discovered was that while he's amplifying a particular DNA strand, he's just incorporating bases into it by a terminal, like by a terminal deoxynucleotidal method. That is, instead of adding a normal nucleotide, just a sec, I haven't included an image for this. So I'll just put, so what happens is in Sanger sequencing is that they're trying to sequence a particular sequence by a terminal deoxynucleotidal transferase. Basically, what happens, you have a nucleotide. That particular nucleotide is not able to, like if that nucleotide gets incorporated, that nucleotide is not able to further sequence further extend the sequence. So what and all we require for a normal PCR? We require the DNTPs, we require primers, we require polymerase, we require a template. That's a normal PCR. So instead of all this, he just added 4 different things, that is DDATP, DDTP, and all for GTP and CTP. So this D stands for dideoxy. That is, when this DDATP is incorporated, another DNTPs cannot extend that chain. So wherever it gets incorporated, the chain gets terminated there. So that was the basic concept. So now, additionally, what they did is, when they enhanced the Sanger sequencing, they radio labeled, initially it was radio labeled, and then they fluorescently labeled all this ATGC, that is the DD tags. So now whenever these get incorporated, you can see that there will be a fluorescent tag attached. So you will get a band that is gray, red, blue, whichever nucleotide is incorporated. So when the amplification is happening, if this gets incorporated, the chain will stop there. In another amplification, say TTP gets incorporated, so it will stop here. So the similar way, you will have multiple small stretches of nucleotide that are ending with a particular fluorescently labeled DNTP. So now what you can do is you can analyze them, you can check the fluorescence that is coming out of these, and you can understand and analyze the bands. From this, you can understand that, okay, after this sequence, this is coming. So this was a very basic understanding of what Sanger developed, and this led to the Human Genome Project. So that is how you were able to understand, and that is how they were able to come up and sequence the entire human genome. So this is what is called as the first generation sequencing. Now, as it was very expensive to do all this in the start, and initially Sanger used radioactively labeled nucleotides, that again is not very preferred to use. And with time, it moved to the next generation sequencing. Now we very commonly use. You guys will be very much aware of Illumina. Illumina is very pronounced in this particular field for the sequencing because it is cheap, it is fast, and it is very accurate. So initially the factors that were there, that is, it is a highly accurate thing, but it had a low throughput. That is, you can only run few sequences in a time because of the longer process, and then you have to constantly keep on checking, and you'll get a lot of number of fragments because it keeps getting truncated in the middle. It is expensive for larger projects because you have to run it again and again, and it is suitable for like sequencing like a small fragment. That for that we can go for Sanger sequencing. But after the Sanger sequencing, then came in picture was the next generation sequencing that started from the mid 2000s. So the characteristic feature for this states that like it has a massive parallel sequencing. So what is massive parallel sequencing meaning here? That is, you have 10 samples. Say you want to get DNA sequenced of 10 different samples. For Sanger, you had to do this like 10 times. That is, if you have 10 samples, you sequence it 10 times. But if you have 10 samples now, in next generation sequencing, you can sequence all of them in one run. So the cost has reduced, the throughput is high, the accuracy is high, and the cost has reduced way low. For what you had to do 10 times, now you're only doing it once, so you have reduced the cost 10 times. So that is why it is more suitable and it is more widely used. And therefore, you can see that there are a lot of projects that are coming up, like the genome-wide studies, the 1,000 genome project, the normal human genome project, all of that started to come up. So initially, it was Frederick Sanger in the early 1970s. And then in mid 2000s, we had the range of next generation sequencing. So in next generation sequencing, there is still, there are also some parts like after Frederick Sanger, then some different sequencing comes. And now at this particular point, we are at higher, like the sequencing continues to evolve with improvements. And we started from a Sanger sequencing, which was very expensive and which enabled the Human Genome Project. And then the read length we generally carried out is 700 to 1,000 because it's for a smaller sequence. And then you have to redo it again and again, and that makes it very expensive. And then we moved in my when we moved into 2000s, we saw the second generation sequencing that is called as the next generation sequencing. Those are the short read sequencing. That is, they will break your entire genome into small fragments, and then those small fragments are getting sequenced. So initially it was Roche, then Illumina, then we moved to the third generation. So now we are in our third generation sequencing. So in our third generation sequencing, we have Oxford Nanopore, we have PacBio. So these are all long read sequencing. That is, they can sequence long reads. So that is, PacBio can sequence reads from 10 to 100 KB and plus. Oxford can lead to ultra long reads, that is more than 1 MB. So if you have a genome of 1 MB, you can get it sequenced in an entire go. That is, the entire sequence can be sequenced in just one go. That's fascinating, right? And similarly, and that is the most comfortable and the most cheap that people generally use is Illumina. And whatever data is generated from Illumina is basically known as the NGS data. So now the NGS data that is getting generated, you can do the NGS downstream analysis. There are multiple of things that you can do that we'll be talking about in like further sessions. So for the time being, is it clear? Do you understand the evolution of these techniques? The starting with Sanger, moving to NGS, all of this, is it clear? Okay. I'll again say, if you guys have any doubts, you can just unmute yourself and ask. So now we'll move on to the next session. Next slide. Okay. So now, what is this next generation sequencing? So I already mentioned that this next generation sequencing refers to a modern technology where you can sequence multiple, like millions or even billions of DNA fragments simultaneously. And why is it important? The major, like the main concept and the main importance of NGS here is its property of massively parallel sequencing. That is you can sequence, like I mentioned, right, billions and millions of fragments in one go. How much it is reducing the cost down, right? If you go for Sanger sequencing, you'll end up paying millions and billions of dollars to get the millions and billions of DNA fragments. But in NGS, you can get it done like in a very less than thousands of dollars. The key advantages for what we can, like, we are using NGS so much. It's high speed, it's high throughput, it is accurate, cost effectiveness, and then because of the cost effectiveness, it is comprehensively and very widely used in the genome wide analysis. And then you can use it for detection of rare transcripts, rare variants, and then it generates an NGS data. So NGS data can include transcriptomics, epigenomics, metagenomics. So transcriptomics here basically stands for the RNA data. That is, we are going to sequence the RNA of a particular organism. Then epigenomics is basically to understand the methylation patterns and all of that, so that you can check like what are the changes or epigenetics that is happening. And then when we move to metagenomics is basically for understanding the metagenome. That is a microbiome environment. You take a soil sample or something and you sequence that and you get a metagenome data. So you understand what all organisms are present in that particular sample that you have taken. So there are multiple NGS platforms that we can use commonly. And I guess I just mentioned before as well. So we have Illumina. Illumina is the most widely used platform, and it produces high accurate short reads. So therefore it is preferred everywhere. Oxford and Nanopore and PacBio both are also highly accurate, but they are a little expensive. No, I won't say little, but they are expensive as compared to Illumina. Therefore, people go for Illumina because of the cost, but Oxford and Nanopore and PacBio, these are a little better versions. They sequence long reads, and then you can detect the entire thing in one go. You don't have to cut short it down to smaller fragments, and then you can understand the isoforms, the genome, the variants, all of that. And for Illumina, there are applications like RNA-seq, whole genome sequencing, ChIP-seq, ATAC-seq. So these are the normal NGS data that routinely gets generated by this Illumina sequencing, and these are widely used in the laboratory for the multi-omics analysis. So RNA-seq here basically tells us the transcriptomic data. Whole genome sequencing will give us the information of the entire genome. ChIP-seq you will get the sequencing of a particular, you guys have to perform, and this is ChIP-seq and ATAC-seq are an experiment. You need to perform first, and then you need to get the sequence, and you get it sequenced here. So ChIP-seq basically tells that on what positions of a DNA, a protein, a transcription factor is binding. And ATAC-seq basically tells that what are the free regions in a chromatin that are available. And like that is not much important for this particular study, but these are also an important aspect of the NGS data analysis. So for our course, we are going to basically focus on the RNA-seq analysis. There's one more advantage of using Oxford and Nanopore and PacBio over Illumina. See, you have a long sequence. Now, one sequence of 100 base pairs, you're breaking down into two, sequencing it as 50 and 15 Illumina. So there are chances that some part of the sequence might get missed, or it can get a little like it can get skipped or something that you might not even know at the later part, until and unless you are doing a very sensitive experiment. But it is cheap, that is why people prefer it. Whereas in Oxford and Nanopore and PacBio, you can see that 100 base pair sequence, you can get the entire sequence sequenced in one go. So that makes it much more efficient and better, because you can understand the entire integrity. So you don't have to assemble, you don't have to do anything, you will get the entire sequence in one go. So that makes it a little more beneficial and useful. But the only thing is that Oxford and PacBio are a little expensive. So labs that have good funding, they definitely go for these things, and lab which have a normal funding, they can go for an Illumina sequencing. That is the most common one and most widely used. So for our analysis also, we are going to analyze the data that is generated by Illumina platform itself. So now the application of NGS is in multiple sectors. It starts from genomic research to functional genomics and moving on to clinical and healthcare. So in genomic research, it is widely used for the entire whole genome sequencing, whole exome sequencing. So what is the difference between whole genome and whole exome here? So a genome, if we talk in terms of transcription, so we know that the entire genome is not copied to form a RNA. There is certain exons, there is certain introns. Only the exons, like only one, the exons are the exons are taken and the introns are removed, right? So the intron is the non-coding part and the exons are the coding part. So that is how a transcript differs from a genome level. In genome, it will have both the coding and the non-coding part, but the transcripts will only have the coding part, that is the exons. So that is how the whole genome sequencing and whole exome sequencing is different. So whole genome, you are comparing or you are analyzing the entire genome. But for whole exome, you're only analyzing the exonic part, that is the coding part. Then you can do variant discovery, that is you can detect if there is any SNP, if there is any deletion, mutation, addition, all of that can also be performed. Then we can do metagenomics. Metagenomics is another branch of this analysis where you are only understanding and analyzing the microbial data. So in functional genomics, we are doing the RNA sequencing. So why are we not putting it in a genomic research? Because now from genomics, we have moved to the functional part where the RNA is getting produced from the genome. So that is when RNA performing any function, that is why we are considering it in a functional genomics. So it supports RNA-seq, single-cell RNA-seq, ChIP-seq, and ATAC-seq. So RNA-seq will tell us the transcription level, that is the expression level. Single cell will also tell the expression level, but it will go on to a very small level of a cell. So in RNA-seq, we are going to understand that, okay, there's one gene. In the entire sample, one gene is upregulated. But in single cell RNA-seq, what we look is that in one gene, that is in one cell of the body, say there are 100 cells that we are sequencing. So out of 100, in one cell, which gene is most abundant, which gene is giving the most transcription. So then we move down to a cell level. And similarly, ChIP-seq and ATAC-seq. In clinical and healthcare, we can understand is as a clinical diagnostics and precision medicine. So clinical diagnostics, you can tell that the transcription levels, the cancer patients and the normal patients, all of that are involved in the clinical diagnostics. And precision medicine is becoming a little more famous and widely used because people are very much focused on like getting a precise medicine for their own cure, for their own disease. So that has enabled this field to grow more and more, because you understand one patient's genome, then you synthesize the medicines for that particular patient, only and only for that patient for which you have analyzed. So this field is very emerging and it has a lot of scope as well in medicine, healthcare, and all of that. So now when we move, so now I mentioned that there was NGS sequencing happening, right? So we have our sample, that is our DNA or RNA. We feed it into the NGS machine. So NGS Illumina that I mentioned, Illumina here is shown as a machine, right? This machine sequences the data. So we put our genome, our DNA or RNA, we put it into the machine. So now after we put the DNA or RNA into the machine, now this machine will sequence it and give us the results in the form of files. That is the FASTQ files. So about the files we will again learn in the coming session. So for just now to understand, that is, there's a machine. You give it the sequences, not the sequences, you give it the sample, that is you want to sequence. So you have isolated DNA, you have isolated RNA, you give the isolated sample to the machine. The machine will sequence everything and then it will give you an output. The similar way we do cooking, right? You have a raw material, right? You have raw material. You will cut and chop everything. You'll put it in an oven, and after you give it to oven, it will give you out a proper prepared food. So here the prepared food is given in the form of files, different sort of files that we'll be studying. So that is what is the most interesting for us, because on those files we do the analysis. So how is this now what our main concept is to understand what is happening inside the oven. You have fed the raw material, right? And you are getting a cooked pie. So now you want to understand what is happening in the oven. Okay, this is getting softened and then this is moving up or something like that. Something is happening, right? So that we need to understand now. So when you have a DNA sample, so you will have a double-stranded DNA, right? As we are doing a transcriptomic analysis, so we isolate RNA from our samples. We convert that RNA into DNA by RT-PCR. Now we have the DNA samples. So now to our DNA samples, we will add adapters. So adapters is basically a sequence that is it is a set of sequences that is two complementary set of sequence. This is one and this is two. So this is known to us. We know the sequence. We don't know the sequence of the DNA because we need to sequence it. But the adapters that we are adding to both the ends of the DNA, we know the sequence of that. So those sequence are known and they're characterized. After you have your sequence of the DNA, you add adapters to both the sides. So these adapters are basically the oligonucleotide sequences that you know. Now you are attaching it to both the sides. Then you do cluster formation. So now what is this cluster formation? So you have a flow cell. So you will have a small chip-like structure, like you will have a small, you guys have seen glass slides, right? A thumbs up will be good if you guys are able to understand. Okay. So you guys have seen a glass slide, right? In a normal glass slide, it is just a small rectangular glass piece. But for this thing, we have a small glass slide with a small, like it is carved a little from inside. So you'll have a small well inside so that the water can flow and it can be retained in that glass slide. So there will be some depression. So in those depressions, and that particular slide is called as a flow cell. So in this flow cell, you will have a small oligonucleotide sequences already attached to it. So as I mentioned before, that we are going to attach small adapters, right, onto the sides of the DNA. So complementary to those adapters, we have attached small oligonucleotide sequences on the flow cell. So this is already attached to the flow cell. We know the sequence of this, we know the adapter sequences as well. So you can see the pink and red are complementary to each other. So you will attach adapters in such a way so that they go and attach. Now what you do is you release your DNA into the flow cell. So now it will go and it will bind to the adapters that are present on the flow cell. And then you let it stay there. As this green is complementary to the pink, so it will bend and it will form a bridge shape structure. So now you have a bridge shape structure and both of them are attached on the flow cell. So now similarly, this is one molecule. All the DNA molecules will be attached like that. Now you remove or wash all the excessive molecules. So whatever is attached to the flow cell will be retained, and apart from that, everything will be washed off. Okay, so now after washing off, you will add primers. So this primer will be complementary to the sequence of the adapter, and then it will go and attach to it and it will synthesize. We will have polymerase. We need to form a cluster. That is, we need to amplify the sequence that we have so that it can be sequenced efficiently. So after amplifying the entire thing, so it will, one second. So after amplifying the entire sequence, so after it gets amplified, you can see the primer is here. Now this is the polymerase. So this will act as a primer and this will keep on amplifying according to the bottom strand. So as it comes here to the green, it becomes an entire double-stranded DNA. Okay. So now this double-stranded DNA is there that is attached onto the following flow cell. So now what we do is we give it a step of denaturation. That is, we'll give a higher temperature and it will cause denaturation. So when the denaturation happen, you know that a double-stranded DNA is broken down into single strands. So initially it was bent and it was connected together, but now it becomes single-stranded because all the hydrogen bonds present between the double-stranded DNA is broken. So now similarly, this pink will again attach to the green, and this green will again attach to the pink, and other bridges will be formed, and again amplification will happen, and therefore it will lead to the formation of a lot of cluster. Is this cluster formation clear? Okay. So now this is how we move to the step of cluster formation. So now after the cluster is formed, now all the excessive molecules are washed away. And after washing, you will have these straight clusters that are standing there in the flow cell. So now you need, one side it is attached to the flow cell. On the other side, it is an adapter molecule to which we know what is the sequence. Now to that adapter molecule, we'll be synthesizing primers and we'll be sending it. So this, you can see this acts as basically the one molecule that is standing on the flow cell. So this is attached to the adapter and that is attached to the flow cell. And the top one is also an adapter, but we know the sequence. So now against this sequence, we are going to construct a primer. So this primer will come and attach here. So now we will provide the DNTPs, that is ATP, TTP, CTP, and GTP that are fluorescently tagged. So now these gets incorporated here. And then as it is getting incorporated, it will give a fluorescence. So if a red is incorporated, it will give a red fluorescence. You will understand that the sequence is T. If A is getting incorporated, you will get a blue or a green fluorescence, whatever the color is corresponding to, and you will get the another sequence. So this will get incorporated based on the complementarity of the template DNA strand. So now, how is this getting incorporated? So this is the adapter molecule that you had, to which you generated a primer and you attached it. And then you have your sequence that you have to sequence. So complementary to a T nucleotide will come that is fluorescently labeled with a blue tag. Okay, so it will come and attach. There's one difference property of these nucleotides that we're attaching here is that you know that to extend a nucleotide it needs to have a 3' OH. So its 3' OH is blocked. That is, as soon as T attaches, it cannot extend further with another nucleotide. That is, it has to wait for some time. A cleavage part will come that will cleave this inhibitor that is attached, and then only it will be able to take the next nucleotide. So that is why this process of amplification here is known as reverse termination amplification. So once you attach a T, everything will be washed off. Then only T will remain there with the blocker, and it will produce a signal that will detect and go and being detected as a blue signal. So we'll understand that it is a T. Okay. So now we'll wash off. Now we'll again give the DNTPs. Now when we give and we'll give a cleavage, this thing will be cleaved. And when this is cleaved, this will be able to take the next nucleotide. Therefore, a G will come. G will again have a blocker. So only G will be present. You can see now the fluorescence from blue has become green. Now you understand that it was initially T, now it has become G. Similarly, it will keep on getting extended, and then you can keep on seeing the difference in the fluorescence. So at the same spot, you will keep on seeing the fluorescence, and you can understand, okay, this is T, this is G, this is C, this is T. And based on this, you will be able to construct the entire sequence of what you want. So this is the basic workflow and the principle of how NGS is doing the sequencing. Is it clear? Do you guys have any doubt in the NGS workflow, like how the sequencing is done? Okay. I hope it's clear. So now we'll move on to the next things. So what is it after we understood? So now we are going to analyze transcription. So there's a word known as transcriptomics. So what is this word transcriptomics basically telling us here? So transcriptomics is basically the study of all the RNA molecules that are present in a cell or a tissue at a particular given point of a time. So this complete set of RNA molecules gives rise to what is known as a transcriptome. And the study of this transcriptome is known as transcriptomics. Similar way, we know that there's proteomics, metabolomics, genomics. So genomics is for DNA, metabolomics is for metabolites, proteomics is for protein. And in a similar way, transcriptomics is for the entire transcriptome. So there's a different cell types. So from all of them, some RNA is produced so that a protein can be produced at different developmental stage, at different environmental condition, at different disease status. In all these different conditions, there will be different set of mRNA that will be produced. So that is generally analyzed, and you can understand that okay. So this is how the mRNA is getting differentiated when the environmental or when the conditions inside a person or a body is getting changed. So now this studying this transcriptome can provide a direct snapshot of what the cellular activity is. That is, if one gene is expressed at a higher scale, that means you can understand that the more number of proteins is formed, and you can understand the cellular activity from that particular analysis. So if you say that there's a gene X is an oncogene. That means some oncoprotein is getting produced. So if gene X is highly expressed, then you can understand that from that particular expression, there is certain protein that is getting produced that is harmful for our body because it's an oncoprotein. So now what is RNA-seq? So RNA-seq is just a high throughput sequencing technique that we are using here to analyze these RNA molecules. So as we are analyzing the RNA and the transcriptome data, that is why it is termed as an RNA-seq. So RNA-seq will allow us to measure gene expression at a higher level, then identify if there are any novel transcripts to detect any alternative splicing events. So what is alternative splicing? When a genome is getting converted to mRNA, there are exons and introns. So when exons and introns are getting transpliced, that is introns are removed and only exons are retained, that part also it can detect and it can tell, okay, these are the splicing events that has happened. So between these two introns, there was an exon that has been removed. That is an advanced version where you can understand the transplicing events. Then you can determine fusion genes, you can analyze transcript structures. There's a never-ending list to the analysis, what and all you can do with the RNA-seq data. So majorly what we use it very commonly is for to understand the expression of the gene level, where we determine the upregulated and the downregulated gene, and we understand that how they are different, how they are getting different and their expression levels. So are you guys aware of what is microarray? Have you guys heard this name before? Anyone, you can just unmute yourself also and answer. Like, have you guys ever heard about microarray? Then I can explain it further in detail if you guys have heard. Okay. So microarray is another technique where you are understanding the expression. So you have a molecule that is what you will do is you will have a chip. So this is how a microarray chip looks like. So microarray also is used in a gene expression analysis. This was the initial ways when we did the gene expression analysis. RNA-seq is an advanced version. So in microarray, it required a predefined probes. That is, researchers can only study the known genes. You cannot detect any novel substance. So in microarray, you had a chip. To these chips, the probes were already attached. So now, when you get your RNA sequences and your RNA-seq data, then what you can do is you can hybridize the sample, that is the sample DNA or RNA sample, onto the chip. If the samples are complementary to each other, they will hybridize and they will show different colors. So if that sample is only present in the cancerous thing, so it shows only red. If it is only present in normal, it shows green. But if it is present and expressed in both of the genome, that is both cancer and normal, then you can see a mixed color that can be seen. But this is at a raw level, like this you can understand, but you can only understand the known ones. So that is when RNA-seq came into the picture. So RNA-seq is a higher and advanced version. So you don't require any knowledge before. That is, see here you need to have probes, right? You need to have some understanding of what sequence or what DNA you're going to sequence. Say you have a sequence of 100 base pairs, you should know some parts of that sequence, then only you can understand it on the probe, on the microarray chips. But for RNA sequencing, if you have a, say, whatever length you have, you don't require any information about it because you are going to attach adapters to it. Those adapters is what you have the sequence, you don't know the sequence for that, and only that you require so that you can directly sequence it and understand the unknown sequence that is flanked by both the adapters. Its sensitivity is higher, it has a larger dynamic range, and then it is basically detecting novel genes. So you get a sequence level information perform an RNA-seq analysis. So we have been talking transcriptomes, genomics, all of that. So now what are we doing? Like so initially we have a sample that we'll collect from humans, we need from plants, whatever sample. Like for whatever understand you isolate. So after RNA isolation, you need to do a live. So what are the steps of live? That is, you will have you need to convert the RNA to DNA because make it more stable and more sequence free. So now you have cDNA. These cDNA should be adapted by both adapters on both sides. We just mentioned before. After adapters addition to both the sides and the DNA in the center, now this becomes a library. So we have a set of sequences, right, which we need to sequence. After attaching adapters, we will send the entire thing for sequence. So NGS Illumina will sequence and it will send me a report. And it will send me a file. So that file is what is important to us now because we need to understand the expression levels from those files. So now there are some designated steps that we generally follow for our analysis. So as soon as we get our files, we need to check each information. So it will tell you how many PCR duplicates are there. PCR duplicates are basically you have one sequence instead of sequencing that thing. A same copy is getting generated of that particular sequence. So that is just coming that for a particular sequence, you have only one read that is coming. For that, multiple copies are getting formed. So that is of no use to us. So PCR duplicates to check the bases, like at the place of A, what is the probability that an A is in and it should be in. So it will give that will move it. After getting, we do the alignment. That is, we have all the scripts, we will align it to a reference. That is whatever. See if human data. If we took a microwave data, we'll align it to a microgenome. And then we'll quantify it. That is after the alignment, you do the quantification. Okay, which gene is expressed more rate, and then you do the differential expression. So this process you can see here right now, I have it as a very cartoonish representation. But this is a volcano plot, and this is a heat map. These two are very commonly used to determine the expression levels and to depict the expression levels in a, and do the functional intelligence for water on online servers against treatment. So this is a very deep, like, understandable. So biological sample, you are happening, you are library, you're doing an mRNA and you are adding factors, so you get a FAQ file. This FAQ file will check the quality, like how is the quality of base and all just explain, mapping, quantification, and analysis. This is a more detailed version that is like want to read. You guys can read it on your own and understand that how are these all steps happening in detail. So now we'll understand all the steps in a little more detail individually because we have to work on that. So from step 1 to step 4, This is just a like pre-processing step. Generally, when you analyze a data as a mathematician or something, you will generally start from a fast cube file. Like your lab will produce the data and will hand you the data analysis. So from there, you have to move on. So this is just understanding a little brief about initial steps that is sequencing. The first step is going to be extraction. How do you RNA and how do you do library prep? So when you isolate the RNA or the content, DNA content from a sample, so you will do the fragmentation. That is, we can't you know that Illumina only does it in a shorter fragments. So you will break down the entire genome, everything into smaller fragments. After fragmenting into smaller pieces, you will go for reverse transcription. That is, you will synthesize a cDNA from an RNA, and then you're going to do an adapter ligation. That is, you will attach the special adapters to both the sides. To these adapters, you know the sequence very nicely. Okay, so you know the entire sequence, and that is why you're adapting it to both the sides. And then you will send it for amplification. Now, this amplification step is where you will move the entire thing to the NGS. So NGS will do the amplification of the sample that you have given, and it will give you the FASTQ files. So now the FASTQ files is the what sequencing output that you are getting. So now these FASTQ files, this is just a basic idea about these FASTQ files. They are going to understand each and every step in a little more detail properly. So these FASTQ files have certain features, and then they have certain standard profiles. So there will be a read identifier. That is, if you have a sequence. So a DNA sequence was given, and then to that DNA sequence, they synthesized a sequence and gave you that, okay, this is the sequence that you were expecting. Initially, we didn't know the sequence. That was an unknown DNA. They sequenced it and it gave me a sequence, A, T, G, C, whatever. Now that is called read, because that read is coming from an unknown DNA. That is what we are calling it as a read. So that read is having a unique identifier. That is, each read will have a unique header or a read ID. You can say, like when you go to college, right? If I'm Anshu, I have my roll number. So that is my ID. Similarly, each read that is getting generated here will have a ID that is called as a read ID. Similarly, it will have the nucleotide sequence. Obviously, in the FASTQ files, whatever your unknown DNA has given a sequence, say A, T, G, C, G, T, that sequence will be mentioned along with its read ID. And there will be quality scores. So the Fred scores, the ASCII codes, so these and all we'll study when we move on to the quality score part. So the basic thing is that quality score is telling that how good my base is. Say, if in a DNA sequence, A is supposed to come. So what is the probability that A is only there? So say if it is a 90% probability that A is there, that means that it is correct, right? It is good that 90% chances are there that A should be there and A is only there. But say if it is saying that it is only 10% probability that A is there, and in our sequence A is present. So you will be like, okay, this is not a good sequence because the probability is less, right? That this sequence, this particular base shouldn't have come here. That is what it is saying when it has a low probability and a low score. That is why we generally try to remove then and modify all of that. That is another quality assessment test that we will be doing. That is different. So from FASTQ files, the major thing that we have to understand here is a quality score. So in just a basic term, I would like to understand, like I would like to make you understand what quality score is. It is just telling how good that base is. Like, is it supposed to be there or it is supposed to be removed? So if it has a good quality score, that means that it is supposed to be there. But if it has a low quality score, that means it is a low quality. The system was not able to detect a good base. Therefore, it just put like that. So therefore, quality score measurement is very important so that our entire sequence is correct and is reproducible. Next is quality control. This is the first step. As soon as we get our RNA-seq reads files, this is the first step that we'll be running. That is, we need to check the base quality of each and every base. We need to check the contamination. That is all the adapter sequences that we added, right? Generally, they are not sequenced because they are put as a primer. Okay. Adapter sequences are put as a primer and only the further sequences is carried out and those only are retained. But still we will check if there is any adapter present or not, or any impurity or any non-essential read, then we will remove that as well. To check the GC content, how much GC bases are there, to check if there are any PCR duplicates, and to check if there are any overrepresented sequences. So all these things will be done in the first check. You will get a files. I'll make you understand how to analyze those files, and you will understand that how we are understanding the quality of our sequencing reads that have come. Now there's one question. Okay, if you have a read, if you have a file, that file shows a very low base quality score and very high PCR duplication. So should you use that file for further for further analysis, or should you drop that and get it sequenced again? It's just normal question, a yes and a no. You guys can write in the chat box as well. I'll repeat the question. If there's a sequence that you got just sequenced, and that particular sequence, it is showing that the base quality scores is low and there is a high number of PCR duplicates. Like one read is getting amplified again and again, again and again. So there are chances that there will be no biologically relevant data in that. And the quality score is also very low. So that data, do you think that that data should be considered further, or that the sequencing should be done again? Yeah. The sequencing should be done again because the quality is not good. And if the quality is not good, then there is no meaning. You will get false results in the next, whenever you do the analysis, you will get false results and it will not be biologically relevant. You will get all the wrong analysis and all the wrong data. So now after you understand the quality, now you move to the next step, that is read alignment. That is, you will map your the raw RNA-seq reads that you got onto your reference genome. So there are multiple tools that you can use for the alignment, that is HiSat2, STAR, Bowtie. So we will generally use HiSat2 commonly. STAR is another advanced version, that is, it is more prominent and it is more accurate, more higher speed, that it requires a larger space as well. So it is a little computationally heavy tool, STAR, but you can use if you, but this tool can be used very proficiently if we have the RNA-seq data on a larger scale. Say we have a very large RNA data, then STAR can handle it very proficiently and efficiently. Bowtie 2 is also used, but it is a little, it is a little less, what do you say, less better as compared to others. So as we know that that mRNA has that splicing events where exons are removed and exons are retained and introns are removed. So Bowtie 2 will align the entire thing. It will not consider the fact that that RNA has this trans-splicing event. So if we do an RNA-seq analysis, we can do the alignment using Bowtie. But when we are doing a trans-splicing, like when we have to understand these splicing events, that is where it is cutting and where it is understanding and where it is joining, for that we generally use HiSat2 and STAR, because both HiSat2 and STAR, they are aware of these splice events, whereas Bowtie is generally a normal aligner that aligns your RNA to your reference genome. Whereas HiSat2 and STAR, they take in consideration of the splicing events, and according to that they align. So that is why they both are considered as better tools. But it also depends on the memory, that is of the system that you are working on. If it has a very good efficient memory, then you can use STAR, but HiSat2 is also a very good tool to do the analysis in the STAR. And Bowtie 2 is a regular tool that you use. It is also giving a good result, but HiSat2 is preferred when you have to understand that there are certain splicing events that are happening. After you understand this particular alignment, you will get files, that is the alignment files. So now in those alignment files, we will do the quantification of expression. So the common tools that are used for the quantification is, so first, what is quantification? So you got the reads, did the alignment. So now you need to see that, okay, this is the sample and how many reads are aligning to that particular sample. So you will have a count table, number of samples and number of reads. So that is called as a count table. So for these count tables, we have so many tools that we generate or we can use to generate. The most common one is we'll use feature counts. So feature counts is very efficient and very highly efficient to give the read counts of a particular chromosome. So it will generate a table of the entire data that will have the read counts and sample. That table can be used further for the expression. So now here another concept that comes is that we generally don't work on the raw counts. We generally do the normalization. Say if from one cell, only 10 read came, and then from another cell, say 100 reads came. So the entire system will become a little biased, and it will choose the 100 cell wala thing, because more number of reads are coming from that, and it will directly say that it has higher expression, even though the cell one also had higher expression. So what we have to do is we have to bring both of them down to a normal scale, that is, say 100, say 100. One is 100, one is 1000. So we'll bring them to 100 scale of 100, and then we'll understand that okay, which one is showing a higher expression. So say now bringing it back to 100, you see that A1 is showing 100 expression, whereas B is showing only 20%. Initially, what we told that, okay, B can be a higher expressing gene, that is false now, because A is a more highly expressed. When you bring both of them down to the same scale, you can understand that which one is a highly expressed gene or which one is a low expressed gene at a very prominent and an accurate level. So to do this normalization, different normalizations. That is FPKM, that is fragment per kilobase of transcript. So this is a method of normalization that we use. TPM is another method that we use. We will be discussing this again in further sessions when we move into much more detail. This is just a basic and an introduction section. So now, apart from the normalizations, there are certain other features, that is the DE-seq2, which we generally use for the quantification and the expression to generate data and plot. We generally use this DE-seq2, HR, and all of this. Limavum we won't be using. We'll be using either of one, that is either DE-seq2 or HR. HR is a little older version, and DE-seq is a little newer version that people tend to use. So now what you will have is, for the gene expression analysis, you will have your RNA-seq data. That data you will use as an input. That is, you say you have taken it from two conditions, condition A and condition B. You need to compare the counts. So after inputting both data, say one is a condition A treated and one is controlled. So now when you have a treated and a controlled sample, you will input, you will do the mapping, and then you will create a gene expression count. So after you create a gene expression count, you can now use the tools like DE-seq2 and HR to do normalization and do the differential expression. So now when you do a differential expression, you will get a plot like this, which will tell that, okay, these many genes are upregulated and these many genes are downregulated. So this plot is generally plotted between the log fold chains. So we'll talk about this also in much more detail, just to understand that this graph basically tells that these genes are upregulated and these genes are downregulated. And you can get a table of a significant downregulated gene and significant upregulated gene. So the examples, like the comparisons that you can done using an RNA-seq data, is between a disease and a healthy individual, between a treated and an untreated sample, between a mutant and a wild type. So these are just two contradictory situations and conditions that you generally use when you have to compare. So if I have to compare myself, so I will compare with someone that is not similar to me, right? If I'm suffering from a disease, I would like to compare myself with someone who is normal. That is how we are comparing and understanding the changes at the transcript level that is happening between a treated and a diseased individual. Next is, after you understand the genes that are highly upregulated and downregulated, now you do an interpretation and an biological enrichment. So there are different websites which you can use for the enrichment, that is gene ontology, KEGG pathways, reactome, and enrichment. So these are the different platforms where you can do the interpretation. So what you're basically doing here is that the genes that you found as differentially expressed, so now you're trying to understand what are the biological implications of those genes, that is, what functions do they perform, and to understand what gene expression will be altered. So if I have gotten a gene, say gene A is the gene that is expressed more. So now I'll do the enrichment and find out that this gene A is what? Is it expressing a particular protein that is responsible for cancer? Is it a particular protein that is responsible for height? What is it? That is what is called as enrichment. And that we'll understand using the biological interpretation after we determine the quantification and the gene expression analysis. So now, what are the applications of the RNA-seq? So when you move on to the application part, there are tons and tons of applications that are present. RNA-seq nowadays, RNA-seq is used in almost everything. So whatever it is, we'll just get an RNA-seq data, and just for the sake of analyzing, we might analyze the data as well, because it has so many features and applications that it is made so far in the field of omics. So in cancer research, if we see, so in cancer research, I've just told, we can understand and analyze a treated and a cancer patient or a normal, say we can use a normal, say two cancer patients are there. One is suffering from leukemia, other is also suffering from leukemia. So now what you can do is you can let's do the sequencing of both of them. One who has undergone chemotherapy, another who has not gone chemotherapy, is just taking some medications. So you can understand the difference in the transcript level at that stage as well. So you can make numerous number of combinations. So you take only one individual, you get a sequencing of one individual before chemotherapy and after chemotherapy. So again, you have two contradictory conditions. So you can make tons and tons of condition and understand the different aspects of a study. So in cancer, it is very widely used. Then it is used in drug discovery, as I told, in precision medicine and all of that. It is very widely used in developmental biology as well, to understand the developmental processes, the stress related things, and all of that. Agriculture, to improve the stress traits and resistance. And infectious diseases is to understand the host and pathogen interactions. So, like if a pathogen is in, see, we take a bacteria, we get the understanding and the sequencing of the bacteria, RNA sequencing. So we see certain genes are normal. But when we get a particular E. coli, which is infected by a phage, a virus, you can see that the transcriptome level is different now. So we can understand by correlating both of them that, okay, from when the virus is getting infected, there are certain set of genes that are getting upregulated and very highly expressed. So those are shown and those are understandable. And then like we can understand, okay, when it is infecting, this protein is elevated, this gene is more expressed more, this gene is therefore involved in the defense mechanism. So this and all can be understood just by the just by understanding that, analyzing the transcriptome data. So now, obviously there are so many advantages and applications, there might be some challenges as well. So sometimes when you do a metagenomics analysis, or when you're doing a lot of or a large number of patient analysis, you can understand that the large data is getting produced. So the data volume and the data space is going to be too much. So you need high computational requirement. So when you have a high computational requirement, it is going to cost a little more expensive to analyze the data, because you need a system that can like take the entire data and analyze it without crashing. So I myself, when I analyze a lot of data, when I do it on a larger scale, like a very, say, 50 GB or 100 GB data, there's often chances that my server crashes or my computer crashes because it is not able to take that much high load. So that is also a very big problem that people face when they have to analyze this data. So they have to make sure that their systems are a little high and running. So this thing should also be taken in consideration, because this is one of the most prominent challenges that are faced by people. After that, it is a biological variability. That is, there is a lot of variations in the data that can make a little difficult to interpret the result. Therefore, we have to make the standards and condition a little standard so that we can understand and compare. So say if we are taking one sample from a 20-year-old boy, and we are taking one sample from an 80-year-old person, obviously both of them are going to be different, and it is going to cause a biological variability. So that is how it becomes a little difficult in that concept as well. Because when we are going to go take samples from the clinical area, sometimes it is missed that from which patient it is coming and all of that. So that can lead to certain wrong results and wrong understanding. So you need to make sure that the biological variability is maintained, and you generally take in consideration all of that, and then only move on to the next step of analyzing. Then we have batch effects. Batch effects introduced during samples that also can lead to confuse. That is, if you're sampling and if you're sampling and sequencing in small batches, that can also lead to certain confusions and some confound analysis. Then statistical analysis can be complex, and it requires a careful experimental design. So we need to have a constant and a very good understanding of the statistical complexes as well, because multiple statistical concepts are used when we are understanding and analyzing the p-values at the later point. That gives us the significance of what our data is trying to tell us. So all of this combined should be understood and should be taken in consideration before we do our RNA-seq data analysis. So okay, this was it for today's session. So in today's session, I majorly covered about the RNA-seq analysis, the RNA-seq part, the workflow, what is RNA-seq transcriptome and NGS. So I hope today's session, I hope whatever I've taught till now, it's clear. Do you guys have any doubts in whatever I have taught, like any point that you didn't understand, you want me to repeat? Come on, guys, you can speak, like you can unmute yourself and you can speak as well.
No, ma'am.
Okay. So that is what is the basic understanding of the entire RNA-seq workflow. So whatever steps we have learned now from the start till the end, we'll be analyzing them separately, like individually as well when we move for the further sessions. And will be performing all of them hands-on. That is, we'll have a FASTQ file, we'll do the quality assessment, we'll do the mapping and all the steps till end. So after the steps is done, you know that there's a mini project thing. So we'll be providing you mini projects. That is, there will be certain few data sets and the aims. So you need to work on those data sets the similar way we will work in the entire pipeline. Then you have to analyze those concepts, analyze the real world data, and then you have to interpret the biological interpretations that you'll be getting from the data. So this is how your mini projects are going to be scheduled. So you will be given a data set. So I'll just stop presenting. So you will be given a data set, and you have to work on that, and you have to replicate the entire pipeline. You have to determine the biological results, and you have to generate a report. So in the report, you will be mentioning the entire thing from the start. Like, you will have an abstract, the introduction, then you will have the material and methods, the protocol that you followed, and followed by results. And after this, there will be a further analysis of whatever you have done by your work that you'll be presenting. So this will be at the later part. First, we'll be mainly focusing on what the RNA-seq pipeline is, and we'll be moving into the further, a little more complex way when we'll be working on the real data set. So is today's session clear? Is it clear that what is RNA-seq and everything? Okay. So I think that is it for today's session, because today's session was all about basics, where we started from the understanding and we moved to the workflow, like an overview of the workflow.
Thank you, Ms. Anshu. Dear participants, I hope today's session is clear. So I could see few thumbs up as well. So yeah, next session is tomorrow. And Anshu, what is tomorrow's session all about?
Tomorrow's session is also a little basic, where we'll be talking about the Linux environment. So these people will be working on the Linux commands as well for the analysis of the RNA data.
Are you going to share them any commands as prerequisite, like whether they're going to work on it or what? Like basic commands.
Okay, so like I want to ask them this question, like, do these people have a Linux environment?
I don't think so, because all of them are using Windows. This was discussed earlier itself. And
Okay.
when it comes to hybrid batch, yes, we have Linux in our laboratory. They can make use of it.
Okay, so can you guys, whoever doesn't have a Linux on their system, can you just raise your hands?
Holy Cross guys, I think you don't have Linux, but we have already given you an installation guide to install
Okay,
Linux,
okay.
right? So have you
Okay.
done with it? No, let's get confirmation.
Yeah.
Have you done with it?
Yeah, you guys
Holy
can just raise your hands, like if you have.
Pasha, Reina, Maria, all right.
Yeah.
Yeah, I think now everyone has Linux environment. Yes, Anshu, because they are using Windows, we ask them to install Ubuntu. So
Okay,
they
okay.
but they haven't installed R. So they
Yeah,
have
our
Linux.
installation and all, I'll give the commands for all of that in the next session. That is not an issue. So now the prerequisite was just Linux. They should have Linux in their systems. That's it.
Yes, all of them has Linux.
Yeah. Okay. Like Bhavani, if you don't mind, like can I have like everybody just introduce themselves once, so that I also know
Yes,
the
yes,
students.
sure.
Yeah.
They can do it.
Yeah. So like everyone, you can, like, one by one, you guys can just unmute yourself and can just introduce yourself, like your name, where are you from, what are you doing, all of that. So it's easier for me also to know you guys, right? Come on, guys, unmute
We should
yourselves.
start with attendance.
Yeah,
I
like
should.
should I tell the names? Like, then only you guys will unmute and speak up. Okay.
Yes, Anshu, that's the only option we have.
Okay, we'll start with Varsha.
Good evening, ma'am.
Good evening.
Ma'am, myself, V. Varsha Veronica, ma'am.
Yes.
Currently pursuing my first MSc Bioinformatics in Holy Cross College, Trichy, Tamil Nadu.
Okay. So you're from Tamil Nadu itself?
Yeah, yes, ma'am.
Okay. Great. Okay, so next we can maybe go with Maria.
Yes, ma'am. Good evening, ma'am. I'm Maria Darshini, ma'am. So I've also been doing the same master's in bioinformatics now. So it's an integrated course, ma'am.
Okay, so you are like been here from bachelor's and master's. So it's
Yes, ma'am.
bachelor's,
Yes,
master's
yes,
integrated.
yes, ma'am.
Oh, okay. That's great. So other people can start unmuting themselves and speak now.
Good evening, ma'am.
Good evening, Girish.
I am Girish Kumar from. I am currently pursuing a master's degree in at Anna University, ma'am.
Okay. So like, okay, you are doing a master's in bioinformatics.
Ma'am,
You are doing your master's in bioinformatics.
computational biology, ma'am.
Computational biology. Yeah, that's
Yeah.
great. Okay, good.
Yeah.
Yeah, good. And you guys can call me like Anshu.
Yeah.
We
Okay.
can. Yeah. Okay. So next.
Good evening, ma'am. I'm Hepzibah, ma'am.
Okay, Ipset, go ahead.
I'm Hepzibah. I'm also from Holy Cross College, ma'am. Now I'm doing my master's, ma'am, in bioinformatics.
Good evening. Tamil Nadu.
Yes, ma'am.
Okay. Next.
Good evening, ma'am. Myself, Wafa
Good evening.
Tapasur. I'm also pursuing my master's degree in bioinformatics. I'm from Trichy, Tamil Nadu, ma'am.
Okay. Let's go.
Hi, ma'am. This is Aisha. I'm also from Holy Cross College, Trichy, and
Okay.
I'm also pursuing my master's in bioinformatics.
Okay, that's good. So all of you are bioinformatics students. I think somebody
Yes.
unmuted themselves. Yeah.
Hi, ma'am. This is Tanushree. I'm from Tamil Nadu, and I'm currently pursuing my master's in bioinformatics at Holy Cross College.
That's good. I think 3 people are still left.
Good evening, ma'am.
Good evening.
Myself, Guru Nindal, ma'am.
Yes.
And I'm currently doing my master degree in computational biology at Anna University, Chennai.
Okay. That's good.
Thank you.
Thank you.
Hi, ma'am. I'm Raina Sharon.
Hi.
I'm doing my master's in bioinformatics, ma'am, from Holy Cross College, Trichy, Tamil Nadu.
Okay, that's good. So you all guys are like batchmates.
Yes, ma'am.
That's good. Okay. So Surya is left, I guess.
Good evening, ma'am.
Good evening.
My name is Ajay Surya. Currently pursuing Masters in Computational Biology, Anna University.
Okay.
Thank you.
Thank you. Good to hear you guys. So all of you are from bioinformatics background. So like all of you had your bachelor's also in bioinformatics or just like masters?
Ma'am, actually, our course is an integrated course, ma'am, for 5 years.
Oh, so it's a integrated full bioinformatics program.
Yeah, so this is our 4th year. Now we are going to 5th year, ma'am.
Okay. That's great. So like I'll introduce. So I'm Anshu. So I'm from Delhi.
Before that, sorry to interrupt you. This Odikra students, they're doing their integrated bioinformatics course. So they have bioinformatics knowledge in the undergraduate. But
Okay.
Anna University guys, only their MTech is in computational biology, BTech is biotechnology. Yes.
Okay,
And also
okay.
we have two from Rajlakshmi Engineering College. Now they are currently not in the session. Again, they are currently pursuing biotechnology, BTech biotechnology.
Oh, okay, okay. So there are people from different backgrounds as well. Yeah, okay, okay. That's good. So I hope you guys are understanding, right? Whatever is happening. So it might go a little over your head in the start, but later you will understand everything. And it is like very interesting to understand like how you can interpret the data and get a biological meaning out of that. So it is going to be interesting. Oh, so I was introducing myself. So hi, I am Anshu. So I'm from Delhi. I have done my bachelor's in microbiology and then I did my master's in bioinformatics and biotechnology from IBAB Bangalore. So it was nice having the first session with you guys. I hope you all understood. So I'll just suggest one thing, that if you guys have any doubt during the session, you can just stop me and ask. I'll repeat the same thing. And you can ask me how much ever times you want to repeat the session. So that is not at all a problem. So that is it for today's session. I really enjoyed talking to you guys as well after the session. Hope you also enjoyed the session. So I think, Bhavani, that's it for today. Maybe we can end the session now.
Fine. Thank you, Anshu. And
Thank
thank
you.
you, participants. Holy Cross guys, feedback link remains the same. It's already there in your WhatsApp group. So kindly fill your feedback for today. And Ananya, City Girish, Surya, and Vahi. Cool. For you guys, I will be sharing it in WhatsApp group. So please fill the feedback form. We're requesting you all to give your genuine concerns, like in case if you find the phase of the session is bit fast, even you can mention the same in the feedback as well. I think on every day we'll be filling the feedback form, and I am requesting you to say your concerns on the same day itself. So we will try to sort it out and try to give better session in the future. With this, let's wind up for the day. You will be receiving session recordings mostly by tomorrow morning. If you have any doubts, kindly keep me posting. You will be getting answers shortly. Thank you all. Thank you, Anshu. Let's meet tomorrow at 6 p.m. India time. Thank you.
Yeah. Thank you, everyone. Bye.
Thank you, ma'am. Bye.
Thank you, ma'am.
Okay, bye.